Good evening, all. First of all, I would like to thank all the organizers for providing me with this platform. So, I am PhD student at RRI, and I am working on to understand the force generation mechanism in living cells. So, I'm trying to understand that how a neuron generate force while it is in growing force. So when we look at the growing neuron, we see this is a bright field image. We see a cell body, and there is a long cylindrical structure which is called axon, and this part is called growth cone. So the growth cone creates lot of membrane nanotubes, and it makes contact with the substrate and advance, which elongates the axon. The length of axon can vary from like hundreds of micron to it can be a few meters long. So it's to study the growth of neuron, it's important to understand how the growth cone generates the force. So if we look inside axon, we see aligned microtubules, and there are a lot of cortical actin filaments enclosed inside membrane wall. So here, this is a image. This is a video of a growing neuron, where I have expressed actin GFP inside it. So whatever imaging is done, we see actin inside the neuron. If I play the video, you see that a lot of membrane tubes are being formed, and then they retract. If there are studies where people have grown neurons inside gel, and one can do traction force microscopy. So there are fluorescent bead embedded in the gel, and the neurons are growing. Then there is a contractile force being generated by this growth cone. So if we track the bead which is coming nearby the any bead coming near to this growth cone, we see load and fail behavior happening. So what we do, we create membrane nanotube using optical tweezer. So when you focus a laser beam, tightly focus a laser beam, it creates a harmonic trap for micron size particle. We can attach those particle to axon. This is an axon, and then we can pull a membrane tube through it. So reason we are doing like that, because when we want to study the force on the growth cone, it's very difficult to catch a membrane tube forming in random direction, and being the growth cone being very dynamic structure, it's very difficult to do any good statistical analysis. So we just artificially create a membrane tube, and then we keep the tube length constant, and just see the force on the bead. So initially we see a constant force, and then after waiting for about a minute, we see a constant load being built up, and then there is a sudden collapse. So we see this load and fail behavior happening in the membrane nanotube. So we just studied these two quantity, wanted to see the distribution of the duration for which the one peak lasts, and how high it goes. So we see it follows exponentially decaying distribution, which tells that the process might be stochastic. So we imaged actin inside these membrane nanotubes. We expressed the green fluorescent actin inside the cells, and then we pulled the these tubes, and when we recorded, we see very highly dynamic motion going on inside the tube, and when we make a kymograph out of it, we see that it's growth and retraction happening. Here, this part is the axon, and the down part is the tip of the tube, and then we see that the actin retracts back. So we tried perturbing the cytoskeleton inside the axon. So actin related proteins are like, whenever there is a contractile force generation, we think of some conventional molecular motors involved. So we try to suppress inhibit those myosin motors, like myosin 2 using blebistatin and Y2762, and then we inhibited myosin 5. We then still we were able to see those force peaks coming, but when we disrupt the actin using latriculin A, which can depolymerize the actin inside the cell, then we don't see those peaks occurring at all. And even when we stabilize the actin, so when we like arrest the dynamic, then also we don't see those peaks coming. And when we deplete the ATP level inside the cell, then also we don't see the active peaks coming. So we conclude from here that this is active process, and the dynamics of actin is important for this force generation to happen. So when we look at the different types of peak, we see that there are small peaks which goes linearly and then collapse. There are peaks which tends to saturate and then collapse. We see there is a crossover phenomena where it grows with one rate and then suddenly the rate changes. The rate change can happen, could be positive or negative. And sometimes we see that collapse happening in multiple steps. So this particular thing gives the hint that there is some discrete number of elements which is generating the force. And we also see that there are like peaks coming on top of another peak. So we thought of a model to explain this. So what we are suggesting that there are actin filaments inside this tube, and the contraction of these actin filaments generate the force. And then we tell that the rate of contraction, it is decreasing linearly with the load on this filament. So there is tensile load on the filament because the bead is trapped in an optical trap, and whenever there is a pulling force generated, the membrane is coming under tension. So each filament can stochastically bind or unbind to the bead, and we take this binding rate to be constant, and unbinding rate we take its exponential function of the load on the filament. So then we can get the force equation, how it will evolve. We get a time scale. The time scale is dependent on number of filaments here. And when we try to fit a peak with this, we can nicely fit it. And now what we can do, that we can discretize this equation, and then we can introduce the stochasticity by introducing the rate by which the number of filament increases is constant, and at some rate it is some filaments getting detached. So that is the exponential function of the load. And when we run the simulation with this, we are able to generate similar peaks, and this is the down is the actual experiment, the up side is the simulated active peak. So for comparison, we see that like in experiment there is a sudden change in the slope. We get that this happens when like we define the model as the contractile rate is decreasing with the load. So there has to be some load at which the contractile rate becomes zero. So that load we can call it as a stall force. So if there is a reduction in if a filament detaches before it has approached the stall force, then we see that there is a sudden change in slope. Otherwise we see the drop in the peak. So we just compare, like partial drop is happening, which we see in experiment like this. We see peak on top of another peak, then there is just single exponentially approaching peak, and then suddenly collapsing. So yeah, so expert is like what could be this model gives us about the some molecular mechanism at microscopic scale what might be happening, but uh, and one more thing what we see in the experiment, that while there is force being generated, we see a helical uh, twist happening in the uh, tube, which gives us hint that there might be some torsional force involved. So there is a protein called actin depolymerizing factor which can bind to actin, and it can change the pitch length of actin. So and this can generate torsion and give rise to the contractile force. So and we see that this is the actin GFP co-expressed with the ADF RFP. We see that they both co-localize inside the tube. So this could be one of the protein which can lead us to contractile force. And what we did, that we introduced constitutively inactive form of ADF and constitutively active form of ADF. When we introduced inactive form of ADF, what we saw that the height of the peak is reduced and the peak duration is increased. So this gives us reason to think that this might be one of the protein which is generating the force. So I conclude with this, that this contractile force generation, this is myosin 2 independent contractile force generation, and there are only few discrete force generating units involved with that can be confirmed with the partial peak drops and the sudden change in the slope while the force is increasing. And we have a model for this, and we think that ADF is behind this force generation. So with this, thank you. I acknowledge a few people here. Susav, which he is in New Zealand right now. He is involved in this project. Sirin is working with us. She helps us most of the biology stuff, and we have some discussion with Jack in Institute Curie. He has helped us to formulate this model. Thank you.
Thank you. So we have time for 2 questions. Is there a question?
Yeah, I want to ask, like, when generally you start with the experiment, so why the initial the static force, like when you take it out, is it because of the time involved, like to take it out, like viscosity and involved stuff?
No, when we initially pull the membrane tube, it's just bare membrane tube. There is no actin inside it. So what we measure is just membrane tension. So after that, the actin polymerizes inside it and then makes contact with the bead, and then we start seeing this force.
Okay, so the time, whatever it takes, is the actin which builds up
this
bridge.
Yeah.
Any other question from the audience?
Yeah, thanks. This was a wonderful talk. I wanted to ask you about this when you mentioned about the beads stuck in gel, right? So you could actually measure like, and they were dispersed all around the gel.
Yeah.
Ensure that all of them would be having like an equal. How do you calibrate their measurements for these different beads, and what is the setup?
Okay, so that was not our experiment, but people estimate the stiffness of the gel, and from there only I don't know, actually there is a distance between bead and the point of force where it is acting. I don't know how it is done, but people do estimate. It's not very clean experiment in terms of like when you want to measure accurate forces, so you need something like an optical tweezer to do that.
So we don't really have much time for one question, but while the speaker is setting up,